Hey everybody from Living Environment. I'm going to teach you briefly about something called codons. This is building on our past lecture, and hopefully you'll have a good understanding of what codons are after this. So I'm going to go over what you should know to start this lecture. Remember, DNA has those 4 bases that pair together. C goes to G, T goes to A. This right here is my DNA. So C and G are always going to pair together, and A and T will always pair together. In RNA, pretty much everything is the same except A and U is the only difference. So A no longer pairs with T in RNA. A only pairs with U. But just to know, if you do see RNA that has T in it, it will still pair with A. You don't have to pair T and U together. That doesn't make sense. T and A still go together in RNA, but when you see an A in RNA, you have to pair with U. So that's what you should know about DNA and RNA. You should also know about protein synthesis. I talked about it in my last video. It's really where you take DNA and turn it into usable proteins, and that happens in 2 steps. The first step is transcription, where you take your DNA and you turn it into that RNA, and that happens in the nucleus. Part 2 is translation, where you take the RNA, it attaches itself into the ribosome, and then specific amino acids are made using RNA, and those amino acids will build into a protein. So this is what you should know by now. We're going to go a little bit deeper into translation, so I just wanted to review some of the steps with you. Remember, the DNA unzips first, the mRNA is made, it's attached right here, or it's kind of created right here. Then it leaves the nucleus, okay, it heads into the cytoplasm first, and then it hooks itself into the ribosome, which you see right here. Then tRNA is made that matches the RNA up and then creates amino acids. Okay, so this tRNA comes in and attaches these amino acids that will grow into a big protein. Okay, and then that big protein will leave and do other work in the cell. Okay, so that's really translation summed up pretty quick. So maybe you're wondering, how does the tRNA know which amino acids to get? And that's a great question. And the answer is codons. So codons are just 3 bases of any RNA sequence. So this is mRNA right here. And if you look at 3 of them, the tRNA will also read them. tRNA is right here, this crazy shaped thing. The tRNA will read those 3 bases, and then it will know, oh, because those 3 bases are, let's say, AAU, it must have this protein, this amino acid that matches it. And then it'll do the same for the next codon. The next codon might be CCU, and then it will get the correct amino acid. So the tRNA is kind of reading those codons to help it know which amino acid to get. Okay, so this little base, 3 bases right here, is a codon. Please ignore my bad drawing. So this chart is called a codon chart, and we use it to identify, we have codons, and then we can use this chart to figure out what amino acid matches that codon. So we kind of do the same job that the tRNA does to match up the mRNA codons to the amino acid. And remember, a bunch of amino acids will build a big protein. So this codon chart helps you to find out which amino acids will make a protein. So let's find out the amino acid for this codon AAG. So the first letter of this codon is A. So we're going to come over here to where it says first letter, and you're going to find A right here. So the codon, or the amino acid we're looking for, is going to have A as its first letter. The second letter is also A. So the first letter is A, second letter is also A. So if you were in class with me, you would use your fingers to kind of trace all the letters till they meet, but for now I'm just going to use my pen to kind of show you. So again, that first letter is A, so you would draw a line until you meet your second letter, which is also A. Okay, so they'll meet in this box somewhere. And then our third letter is G. Over here we have our third letter options. So you're going to look for G. And remember, it can only be in this row. So this is the G we're looking for. So our codon is right here, AAG. The amino acid it makes is LYS. LYS is really just a short name for an amino acid called lysine. But to keep it simple, we just call it LYS. So we found the amino acid that pairs with the codon AAG. So that is how you use the codon chart. So we have found our amino acid based on the codon. So now our goal is going to be to use the codon chart to determine an actual whole amino acid chain. We just did it with one amino acid before. Now we're going to build a whole chain of amino acids that will eventually become a protein. So we have the DNA here. It's this right up here. And it's already been translated into RNA for you. So this will be one codon, this will be another codon, CCA, ACG is your other codon, and then UGA. So all 4 of these are the codons we're going to put on the codon chart. So just a reminder, codons can only be RNA, and you'll see that because there's only these letters on the codon chart. There's no T's on the codon chart because only RNA is on the codon chart. Alright, so we're going to come up with 4 amino acids here. We have AUG as our first codon we're looking at. So we're going to start with our first letter over here, which is A. Okay, our second letter is U. So we're in this box, A, we're in this row, I'm sorry, and then U will keep us in this little box here. Okay, one helpful thing you can do is, at this point I know my last letter is G, so I can literally just look for AUG in this chart. I don't have to go all the way over to the third base and draw that line. It is kind of helpful, but you can also just look for AUG on the chart. You'll find it. If you're really, really lost, you could just be like, I forgot how to use this chart. I'm just going to look for this codon AUG throughout the whole chart until I find it. That's one option. But you can see that the amino acid is called MET. MET is actually what's called a start codon. It always starts an amino acid chain. So you need MET to kind of get the party started when you're building an amino acid sequence. The next one is CCA, which is weirdly the name of my high school. So the first letter is going to be C, so that keeps us in this row here. Okay, we're going to go until we meet the next letter, which is also C, so that puts us in this box. Okay, and then the last letter is A. Bam, CCA. All of the amino acids, all the codons here make the same amino acid, but still it makes pro. So we have MET and now we have pro. So those are our 2 amino acids. Alright, so we have MET and pro, and now we're going to go looking for our third codon, which is ACG. A is our first letter, so we're definitely going to be in this row here. We'll drag until we meet our second codon, which is C. So that puts us somewhere in this box here. And then the last letter is G, which brings us to ACG, and that protein is THR. So that's our third one. Hopefully you feel like you're getting the hang of it. It's so much easier to do this in person, but for our purposes, we got to do it like this. So now we're going to find our 4th amino acid. We have 3, MET, pro, and THR, and we're going to look for our last one based on this codon here, UGA. So the U is up top. That's our first letter. We're going to drag it to our second letter, which is G, and then our third letter over here will bring us to UGA. And the protein that, or the amino acid that is made, is called stop. So this literally means stop. So at the beginning I said that the protein, the amino acid MET, actually means start. So you have your start amino acid, and you have a few others, and then you have a stop amino acid. This kind of lets the protein know it's done being made, or else the amino acids would just build up, and you'd have infinite amino acids. So you need a stop amino acid to kind of end the chain. So you need a start, and then you need a stop. So it's important to know that. They won't always end with a stop like on the regions, but for our purposes, I just wanted you to see a start and then a stop. Okay, so this is just a summary. So remember, we have transcription, which is where you have DNA gets turned into RNA, which is what we have right here. Then you have translation, which is where the codons, which we just worked with, are then turned into amino acids. So we just practiced for a really long time what's happening here. The codons being turned into different amino acids that form a chain. So that is the process of making a protein. And remember, this chain will build into a much larger protein. So that is how proteins are made. I know the codon chart seems a little confusing, but hopefully we'll get to practice together if school reopens. If not, just keep coming back to this video and I'll post some other resources, and you're going to get more practice with today's assignment as well. Alright, goodbye everybody!